Hi, welcome to our genetics class. For the second lecture, we're going to discuss mitosis, meiosis, and a little bit of the cell cycle. So it will be a review for some of you. There'll be some new material for others. But most of all, I hope you enjoy it, you get something out of it, and as you're watching this lecture, try to think of some questions for me for question time at our next meeting. Enjoy. Today, we are going to be covering the subject of cell division, and in particular, how DNA is reproduced during this process. And there are two types of cell division: mitosis, which involves a duplication of the cell and creating the exact same number of chromosomes in each of the daughter cells, and meiosis, which is a special form of cell division that happens in the germline. And this leads to haploid gametes. But first, I'd like to review a little bit about the types of cells, prokaryotes versus eukaryotes. Then we'll get into the cell cycle. And then after that, we will discuss mitosis and then meiosis. As you may remember from Bio 101, there are two types of cells: prokaryotic and eukaryotic. Prokaryotic cells contain a cell wall which encases a plasma membrane and DNA that is in a nucleoid. The DNA in the nucleoid, of course, contains the entire genome of the prokaryote. In order for this genome to be expressed, RNA polymerases bind to regions near genes and cause their transcription into mRNA. The mRNA travels to the ribosome, where it is translated into proteins. And this, of course, is the central dogma of molecular biology, and this holds true for both prokaryotes and eukaryotes. Prokaryotes are unicellular organisms. They're commonly known as bacteria. There are two types of these organisms: eubacteria and archaea. Eubacteria and archaea are actually distantly related to one another. Archaea actually have more in common with eukaryotes than do eubacteria, particularly in how they transcribe genes. From an evolutionary perspective, then there are three major types of organisms: eubacteria, archaea, and eukaryotes. Prokaryotes usually have a single circular genome. The DNA is not enveloped by any membrane, and there are no membrane-bound organelles such as mitochondria or chloroplasts. Eukaryotic cells are much more complicated. Here we have an animal cell on the left and a plant cell on the right. These cells contain a nucleus. Inside the nucleus is all of the chromosomes that make up the genome. These chromosomes are encased by a nuclear envelope. There are mitochondria in both of these cells, and these organelles are important for ATP production and the redox homeostasis of the cell. Chloroplasts are unique organelle that are found in plants, and these organelles are the site where photosynthesis takes place. The endoplasmic reticulum is important in protein and lipid synthesis, and parts of the endoplasmic reticulum are decorated with ribosomes, the site of protein translation, and this is the rough endoplasmic reticulum. There's other parts of the endoplasmic reticulum, sometimes people just call it ER, that is smooth, and this is more associated with lipid synthesis. The Golgi apparatus packages proteins into membrane-bound vesicles before they are sent to their destination in the cell. And it resides at the intersection of the secretory, lysosomal, and endocytic pathways that these cells possess. It is important for processing proteins for secretion, and it also contains a set of glycosylation enzymes that can attach sugars to proteins as they are processed by the Golgi. So let's compare prokaryotes and eukaryotes. Prokaryotes are unicellular, whereas eukaryotes are multicellular and also unicellular. The examples of prokaryotes are the eubacteria and the archaea. Examples of eukaryotes are animals, plants, both of which are multicellular, and single-celled organisms, yeast. Prokaryotes do not contain enveloped organelles, whereas eukaryotes do, such as mitochondria and chloroplasts, as discussed before. Prokaryotes do have a cytoskeleton, but it is not as complex as the cytoskeleton in eukaryotes. Prokaryotes do not have a nucleus. Their DNA is not enveloped in a cell membrane. But for eukaryotes, this is one of the defining things about them, that their genome is stored inside the nucleus, enveloped in a membrane. The genome of prokaryotes is circular, and for eukaryotes, it's linear. Prokaryotes usually have one chromosome, whereas eukaryotes have multiple chromosomes. Prokaryotes have one set of chromosomes. That means that they have one copy of each gene. Eukaryotes have two or more sets. That means they have multiple copies of each gene. So prokaryotes are called haploid because they have one set, one copy of each gene. And eukaryotes are mostly diploid, so they have two copies of each gene. Each copy is on a homologous chromosome, and I'll explain that a little bit later today. The amount of DNA in the genomes of these two types of organisms is very different. In prokaryotes, you have a relatively small amount of DNA in their genome, and for eukaryotes, you have a relatively large amount of DNA in their genome, and we discussed that in the last lecture. For example, E. coli is 4.6 megabases, and the human genome is over 3 gigabases. Prokaryotes have a very small amount of nucleoprotein, that is protein that is associated with DNA. They don't have histones, and they have some proteins that are useful in compacting the DNA. Archaea are different from eubacteria and are more like eukaryotes, so they have histones that help compact their DNA. And of course, eukaryotes, their DNA is complex with many histones, and the complex of DNA and histones is called chromatin. The eukaryotic chromosome has this general organization. It is a linear structure that contains telomeres on either end and a centromere in the middle. The centromere is an attachment point for spindle microtubules, and these attach to the centromere and are responsible for moving the chromosomes during cell division. The location of the centromere is also used for chromosome classification because the centromere position along the chromosome changes based upon the chromosome that you're looking at. The telomere are at the tips of the linear chromosome, and they stabilize the chromosome ends. And the length of these structures, these telomeres, are important in aging and in cancer. I want to talk a little more about how the eukaryotic chromosomes are constructed. So you can see here that the chromatin consists of DNA wrapped around histone proteins. And this helps solve this ultimate storage problem where you have many feet of DNA that you have to compress into a very tiny nucleus. So to do this, DNA is wound. These histone proteins And the winding of DNA and the overall architecture of the chromosome is also important in controlling gene expression. And we will be discussing that later on in the course. So now I'm going to show you a very nice video from the Howard Hughes Medical Institute on chromatin, and it also goes into a little bit on cell division. So I am going to let the narrator of the video take over.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So I'm going to focus now on prokaryotic cell reproduction. This form of cell division is simpler than what goes on in eukaryotic cells, but it shares some of the same principles. So I think it's best for us to start with the simple version of cell division and then move on to the more complicated cell division that occurs in eukaryotes, which is called mitosis. So first, the DNA replicates, and then during this process, the bacterial cell is also growing. The 2 chromosomes begin to separate, and then you can also see a cleavage furrow forming in this cell. The cells divide by binary fission. Prokaryotic cells reproduce by this doubling, by this binary fission. And for example, E. coli will double every 20 minutes in the laboratory, as long as they have enough nutrition to grow optimally. And now we're going to turn to a video by Dr. Brett Finley, also from HHMI, about cell division.
Bacteria doubles, 2, goes to 4, goes to 8, goes to 16, goes to 32. And in 24 hours, we have 2 to the 72 microbes sitting there. They obviously have the capacity to divide very, very fast. And they continue to grow. And then we're left with basically overrun with bacteria. So you see that, you say, well, how come we're here today? How come microbes just haven't taken over the world? Well, this is in a lab situation where there's lots of nutrients, they can grow quite fast. And the ability to grow like that is really when nutrients are when there's lots of them. Inside the intestine, it's actually tough slugging. E. coli only divides once every 24 hours in there. That's because it's battling out with all these other neighbors for the food, so it doesn't grow very fast. And there's limiting factors. Unlimited growth takes unlimited energy. This world does not have it.
Thanks, Dr. Finley. Now we're going to turn to eukaryotic cell division. Eukaryotic cell division shares some of the same principles of prokaryotic cell division, but it is more complex. DNA must be replicated and then followed by separation into 2 daughter cells. But it is more complex, and why is that? DNA is separated from the cytoplasm by a nuclear envelope in eukaryotes. There are two sets of chromosomes, as you can see above, with the human set of chromosomes, that you can see that there are 23 pairs of chromosomes above, each containing potentially different alleles of the many genes that are on each chromosome. Each pair of chromosomes is known as a homologous pair. Homologous pairs are nearly identical except for the sex chromosomes. And humans have 23 pairs of chromosomes, which adds up to 46 total chromosomes that must be replicated during cell division. Eukaryotic chromosome is replicated, and the two copies of the chromosome remain attached at the centromere. It is at the centromere that the chromosomes will eventually separate during cell division. Spindle microtubules attach to the kinetochore and then eventually pull these two chromosomes apart. Before I tell you more about cell division in eukaryotic cells, it's important for us to review the cell cycle. The cell cycle is the life story of a cell. It is critical for efficient transfer of information from a parent to daughter cells. A new cell metabolizes, grows, replicates DNA, and then divides. There are two major phases, interphase and M phase. During interphase, cells undergo an extended period of growth and development and is divided into three subphases, G1, S, which is for synthesis, and G2. After G2, cells begin the process of mitosis, or M phase. M phase is this period of nuclear and cell division. It includes mitosis, the process of nuclear division and chromosome replication, and cytokinesis, the process of cytoplasmic division. Now we're going to go through the stages of mitosis. At the beginning, we're in interphase, and this is a phase before the chromosomes have condensed, and it's during this time that the DNA is replicating. Chromosomes are relaxed. They're only visible as diffused chromatin. The nuclear membrane is still visible in interphase, and each chromosome has already duplicated during the S phase. M phase, or the mitotic phase, cells have passed the G2M checkpoint, and these checkpoints, there's a whole regulatory network of proteins that control where a cell is in the cell cycle. The M phase is a period of active cell division. It has two major events: the nuclear division, mitosis, and the cytoplasmic division, cytokinesis. Mitosis is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. How long does the cell cycle take? The time spent at each phase varies with species and cell type. However, the shortest time is spent in the M phase. As you can see here, interphase lasts 23 hours out of a 24-hour cell cycle, and mitosis only takes about 1 hour. The phases of mitosis that are the longest are prophase and telophase and cytokinesis. Metaphase and anaphase happen relatively quickly. So these are the hardest things to observe. Now I'm going to show you an HHMI video showing cell division in the C. elegans embryo. The image on the left, you will see, shows the morphology of the cells. The image on the right shows GFP fluorescence. And what is marked by GFP are the centrioles and the chromosomes. The male and female pronuclei will migrate towards each other. the chromosomes will condense, then the nuclear envelope breaks down, the chromosomes form a metaphase plate, then anaphase segregation occurs, and then the chromosomes begin to separate, and then telophase and cytokinesis occur, and then you have two cells. The nuclei come together, the chromosomes are condensing, the nuclear envelope is breaking down. You see the chromosomes come together on the metaphase plate. They separate during anaphase, telophase, and we have cytokinesis that is beginning to occur there. So let's go through these steps of mitosis one by one. During prophase, the nuclear membrane is visible, the chromosomes condense, chromatids are visible under the microscope, and the mitotic spindle grows out of the centrosomes. During prometaphase, the next step, the nuclear membrane disintegrates, spindles enter the nuclear region and make contact with the chromosomes. Spindles anchor at the kinetochores, which are pictured here. The kinetochore is a complex of proteins that had previously assembled on the centromere that allow the attachment of the spindle microtubules to the duplicated chromosome. During metaphase, the chromosomes arrange themselves in a single plane between the two centrosomes, and this is called the metaphase plate. The microtubules radiate outwards, and during anaphase, the sister chromatids separate and move towards opposite spindle poles. Movement is actively controlled by the chromosomes and by molecular motor proteins in the kinetochore. After separation, each chromatid is considered a chromosome. And then we have daughter chromosomes that are in two nearly separated cells, but they're not quite separated yet. During telophase and cytokinesis, the daughter chromosomes arrive at the poles. The tubulin that made up the spindles is depolymerized. The nuclear membrane reforms around each set of the chromosomes, and this produces two separate nuclei within one cell, at least for the time being. The condensed chromosomes relax, the cytoplasm divides, and then this is cytokinesis. Now we have two separate cells. In some cells, telophase and cytokinesis occur simultaneously. And then we're back at interphase. This is a summary of the chromosome number and DNA content during mitosis. The number of DNA molecules doubles during S phase, so you have 4 chromosomes, each with 2 attached chromatids. These chromatids eventually separate at anaphase, where the chromosome number briefly becomes 8 until the cell divides. This has produced genetically identical progeny that end up with the same number of chromosomes that the cell starts with. We have gone from diploid to diploid. And now I encourage you to take a break. We are going to talk about meiosis next. Go get a glass of water, something else to drink, walk around a bit, allow yourself the time to absorb what you've heard in the lecture. Everyone says this helps, so I'm going to advise this to you guys. Sexual reproduction increases genetic diversity in species. Here I'm showing 2 flies mating face to face in midair. Today, I'm going to use the example of the model organism C. elegans to illustrate some of the aspects of sexual reproduction, fertilization, and the early cell divisions that occur during embryogenesis. Dr. Barbara Meyer will be discussing these subjects in these next two videos.
I'm going to show you a video of males and hermaphrodites mating. Here we have the male rubbing up and down the hermaphrodite body. You can see that he's searching all over for that vulva. He oftentimes isn't so smart in how he does that. He looks around for a long time. What you'll be amazed to find is if you watch worms for a long period of time is this is what they always do. And in fact, this is genetically programmed. Every single male does the same thing. And this genetic program can be studied in great detail. Now, if you look right now, that male is successfully finding the vulva, and he's beginning to inseminate the hermaphrodite with his sperm. It's a video. First, you're going to see ovulation. You're going to see the oocyte here passing through the spermatheca, becoming fertilized to become the embryo. Soon you're going to see a video in which the oocyte nucleus and the sperm nucleus come together. You can see the chromosomes condensing as the nuclei come together. You can see that as the chromosomes condense, the chromosomes come together, align together. This chromosomes from the hermaphrodite mother and the chromosomes from the male partition. They then resegregate, and you can see cell division. So once you see cell division, this embryo continues to be growing inside the hermaphrodite. It grows for quite a while, and then what happens is that hermaphrodite lays an egg. And you're going to see the next video of egg-laying hermaphrodite. Next video, please. Watch quickly. Watch where that arrow is, because that embryo is very small. It's a 20th the size of the animal. That happened very quickly, so we're going to run it back in slow motion so you can see the egg coming back through the embryo to the hermaphrodite. There you go.
Sexual reproduction involves two processes, meiosis and fertilization. Meiosis is a different kind of cell division that occurs only in the germline of an organism. It is very different from mitosis, in part because its M phase has two stages, Meiosis I and Meiosis II. The interphase is similar. You have an extended period of cell growth and development. It's divided into three phases: the first growth phase, the DNA synthesis phase, and the second growth phase. However, the dividing phase, this M phase, is different. There are two nuclear divisions, Meiosis I and Meiosis II. These reduce the chromosome number to half, produce daughter cells that are genetically different than the parents, both because they are haploid and also because of the process of recombination or crossing over, which we will discuss. Here's an overview of meiosis. During meiosis I, there's a reductive division. The homologous chromosomes pair. The chromosome number is then cut in half. These stages include a prophase I, a metaphase I, anaphase I, and a telophase I. During meiosis II, there is a mitotic division, and this is an equal division, and the stages include another prophase II, metaphase II, anaphase II, and telophase II. And I'm going to go through all of these steps. Prophase I happens during meiosis I. Prophase I can be divided into different stages. In the middle of prophase I, the nuclear membrane is visible, the chromosomes condense. In late prophase I, the chromatids are now visible under the microscope, and spindles grow out of the centrosomes that contain the centrioles. During late prophase I, homologous chromosomes pair, crossing over occurs, and this is when recombination occurs, and then the nuclear membrane begins to break down. During prophase I, an important process occurs, and that is crossing over. or recombination of the chromosomes. During the leptotene phase, chromosomes contract and become visible. During the zygotene phase, homologous chromosomes pair and begin synapsis. The chromosomes also continue to condense. During the pachytene phase, the chromosomes become shorter and thicker, and the synaptonemal complex develops between homologous chromosomes. This complex facilitates the crossing over, or the recombination, of the homologous chromosomes. In diplotene, centromeres of the paired chromosomes move apart. The two homologous chromosomes remain attached at the chiasma. During diakinesis, the final stage of prophase I, paired chromosomes shorten and thicken and the nuclear membrane degenerates, which prepares the way for metaphase I. During metaphase I, the homologous pairs align along the metaphase plate. The microtubule spindles from each pole attach to one of the homologous chromosomes. This allows the chromosomes to be pulled apart during anaphase I, towards the two poles of the cell. During telophase I, the chromosomes arrive at the spindle poles, the cytoplasm divides, and we get two cells with half the number of chromosomes. Between Meiosis I and Meiosis II is interkinesis. The nuclear membrane forms again, the spindles break down, the chromosomes relax. However, in some cells, they go straight from cytokinesis to Meiosis II. Meiosis II is very similar to mitosis, but the products that you end up with at the end are very different. Meiosis II has four similar stages: prophase II, metaphase II, anaphase II, and telophase II. And I will describe these phases now. In prophase II, the chromosomes recondense, the spindle forms again, and the nuclear envelope breaks down. In metaphase II, the chromosomes align on the equatorial plate, and sister chromatids face the opposite poles. This prepares them for separation in anaphase II, where the kinetochores separate and the sister chromatids are pulled apart. Each chromatid is now a distinct chromosome, and in telophase II, the chromosomes arrive at the spindle poles, the nuclear envelope forms again, and after cytokinesis and the division of the cytoplasm, you get four products. Each of those products is a haploid, not a diploid. And this is the major difference between meiosis and mitosis. There's also some other differences which we are going to discuss. So what are the consequences of meiosis? Meiosis comprises two divisions, which ends up yielding 4 cells. The chromosome number is reduced by half. You go from a diploid cell, 2n, to a haploid cell, n. The products are different from the parental genome, resulting in greater genetic variation, and that's one of the goals of meiosis. There is genetic exchange between non-sister chromatids, and this happens during recombination or crossing over. And there's random distribution of the final chromosomes. Let's say crossing over doesn't happen. You have a parental cell here. This is a parental germ cell, and it has a chromosome from the mother in red and the father in blue. The mother has allele capital A and allele capital B, and the father has allele little a and little b. During meiosis I and II, let's say there's no crossing over or no recombination occurs at all. These chromosomes are unaffected by the synaptonemal complex. the products you will get are 2 maternal haploid cells and 2 paternal haploid cells. So these closely resemble the genotypes of the parents. Let's imagine crossing over takes place, and there is exchange between non-sister chromatids that gives rise to genetic variation. So let's review how that occurs. If you have a homologous pair of chromosomes, the red from the mother, the blue from the father, you go through the stages of meiosis, you get S phase, DNA synthesis, and you produce sister chromatids. This is a duplication of each of the chromosomes, but is still connected at the kinetochore. Then crossing over occurs. This is done by the synaptonemal complex during prophase 1, and non-sister chromatids recombine their DNA. As crossing over continues, you have your prophase 1 crossover that already occurred. You complete meiosis I, the homologs separate, and you get 2 different cells, each with a recombinant chromosome. But only one of the chromatids is recombinant, you can see, in each of these cells. Then meiosis II happens, and this produces 4 different products. A product that closely resembles the maternal genome, a product that closely resembles the paternal genome, the genome from the father, and 2 recombinant chromosomes that are a mix of the mother and the father. Another way to create genetic diversity during meiosis is random assortment. This is the random distribution of chromosomes which independently assort. Let's say you have 3 pairs of chromosomes. DNA replication would occur, and the chromosomes would randomly assort. This would lead to a possibility of 8 different gametes. And let me show you how. You would get 4 different arrangements of the chromosomes, potential arrangements of the chromosomes during meiosis I. And that would cause 8 different types of gametes to be formed by the end of meiosis II. Now, if you consider the human genome with its 23 pairs of chromosomes, that leads to over 8 million combinations. So you can imagine the genetic diversity that exists when you have a larger number of chromosomes. Overall, mitosis and meiosis share similarities. They're both methods of cell division, but mitosis's goal is to produce 2 daughter cells that have the exact same genomic information as the parent cell. So it's trying to make a perfect copy. During meiosis, you want to go from a diploid 2N genome to a haploid genome, and because meiosis is associated with sexual reproduction, you want to increase the genetic diversity of the gametes. And this is achieved by recombination, which takes place during prophase 1 of meiosis. Meiosis eventually leads to the production of 4 gametes from 1 cell. So let's discuss meiosis in humans and learn a little bit about how we make our gametes. So in the male, we undergo spermatogenesis. There are several steps. The first step is the division of the germline stem cell, the spermatogonium, which of course is 2N. It is a diploid cell. Meiosis I begins in the progeny of the division of the spermatogonium. This is the primary spermatocyte. And this is the cell where meiosis I begins. During the process of meiosis, you produce these secondary spermatocytes, that are 1n. By the time meiosis II is completed and cytokinesis is complete, you have 4 equally sized cells. These are the spermatids. These are all haploid, and they mature, their cell structure reorganizes, and they become sperm. In female oogenesis, you also have a germline stem cell, the oogonium, and this self-renews and occasionally produces primary oocytes that undergo meiosis I. At the end of meiosis I, you get an unequal division of the cells, unlike what happens in the male. So you get a larger secondary oocyte and a smaller first polar body. At the end of meiosis II, this unequal division happens again, and then you produce the ovum, which is 1n and haploid, and then the second polar body. This uneven cytokinesis ensures that there is sufficient products within the ovum to facilitate the growth of the embryo when it's fertilized by the sperm. And this happens during fertilization. Then you form a zygote. That's once again 2n. And then with a little luck, this zygote will develop into an embryo. Well, I hope you've enjoyed our discussion of the cell cycle, mitosis, and meiosis. Please think of questions for question time, and I will see you next time when we're going to discuss Mendelian genetics.